the title of this section, it's similar to what we've been talking about, but here again, we're dealing a little bit more, it's still very conceptual and theoretical materials, but it gets a little bit more mechanistic. Does that work? Yeah, it gets a little bit more mechanistic, and so let's do it. So some key terms. Human disease network. So the last term we discussed on Wednesday was the diseasome, I believe. If that wasn't the last, it should have been the last. And so we mentioned, or I mentioned when we were talking about diseasome, what we're doing there is just trying to illustrate how various diseases relate to other diseases. We are essentially building an interactome for diseases, where diseases are the nodes. The nodes are not proteins, the nodes are not RNAs, the nodes are not genes, the nodes are diseases. And so what we're doing is we're illustrating by our edges those interactions between diseases. We are illustrating the relationship between diseases and then also making some predictions about how they're related. Well, so what a human disease network does is it takes this basically to the next level and attempts to basically create a map in which you can predict comorbidity. In that you know which diseases tend to be comorbid, that is, if you have one, you typically are going to have the other. And you use these networks to make predictions about what you're going to see as this person's pathology continues, right? So if somebody comes in with like particular genetic signatures for a genetic illness, and then they have some of the environmental triggers for it as well, and so you're pretty certain that at some point they're going to develop this genetic illness, and then you use these disease networks to anticipate other genetic illnesses that are likely to appear at some point. And so when you start to develop your preventative measures, or even if somebody's already coming in with a particular genetic illness like type 2 diabetes or I don't know, some type of atherosclerosis, that you can anticipate other issues that are going to be likely. And not just based on probability, but based on this is a real molecular interaction between the mutant protein here and a mutant protein here. Listen.
Wouldn't that also be, I was going to say, what's not just a, what was the word you used? Predicted.
Right. That's just not based on probabilities. Right. That's not just based on probabilities. Because when we build these networks, what we do is we illustrate very significant mechanisms that connect these nodes. Right. So when we're building one of these graphs or one of these maps, I argued that a map is a better term to describe these things we build for interactomes, although we do the same thing for diseasomes. These nodes represent actual biological components in an interactome or an actual disease in a diseasome. And then our edges that connect these nodes, they represent something that is real, that is biologically meaningful, right? Some interaction between proteins or some interaction between an RNA and the expression of a gene, right? They're representing something that's actually real and tangible. Versus if you're just operating on probabilities, you basically are in some ways treating somebody for something that may never happen, and you aren't treating it in the best possible way. Whereas if you understand why two genetic illnesses are comorbid, and we'll talk more about this in a little bit when we talk about why we answer the question of why our disease is comorbid. These networks attempt to really illustrate how they're connected so that when you develop your treatments or your preventative measures, you are doing so in the least invasive, most effective way possible. Okay? Whereas if you're just operating on probability, I'll give you an example. let's say somebody comes in and they are 57 years old and they are 85 pounds overweight and they have type 2 diabetes, right? And you can be like, okay, there's a pretty good probability that you are also going to develop atherosclerosis and have some other comorbid diseases, right? That's one thing. And you're like, okay, so what should we do to treat that? Should we use, pharmaceuticals to try to prevent the buildup of plaque? Should we just go in and I don't know, just go in with some stents and just put some stents in already, even though they're not needed yet? Or do you instead say, okay, here's the link between type 2 diabetes and atherosclerosis. Here's the link genetically. Do they have that particular marker? Here's the link environmentally. Do they have this particular environment? If somebody comes in that's 57 years old and 85 pounds overweight, pretty good chance they've got the environmental triggers already. But also you look at these genetic, and then it allows you to basically plan how you're going to prevent that in a more targeted way.
Is there any way they could develop it without either of those genetic?
Oh, sure. Yeah.
So the probability still might be helpful, because they might have either the genetic markers or the environment, but for some reason it happens because of some other reason we didn't know.
Oh, sure, but that's why we would do screenings, right? We'd screen for environmental triggers and for genetic signatures, genotypes that correlate significantly with those genetic illnesses. Yeah.
But okay.
But sure, I mean anything is possible. All right, Phenotypic Disease Network. Phenotypic Disease Network is a visual representation of comorbid disease that when you build it, doesn't take anything into account except comorbidity. And so it doesn't take, it's not really, it's not mapping genes and building like a disease map for, several genetic illnesses simultaneously. You can look at it as like this phenotypic disease network. It's just going out and it's illustrating all comorbidities and how often they are. And you can do it for different people groups, right? So you can do these phenotypic disease networks only for people over the age of 60. Or you can do these phenotypic disease networks only for people from this particular region. But basically what you're doing with this is just illustrating all of your comorbidities so that you can from there develop more specific detailed studies to try to ascertain the reason and the way in which diseases are related. Okay, so this is basically where it starts, where you would start. We'll talk a little bit more about these in a bit, but these basically start as retrospective studies on millions of patients, okay, where they basically just get medical records from millions of patients and look at all of these instances and prevalences of comorbidities. All right.
Can you repeat real quick the purpose of this?
To begin the process of developing studies to understand the relationship among genetic diseases. Yeah. Yep. Protein-protein interaction. This is something we've seen before, but it's important to talk about in this context. Remember, last time we actually finished off with a diagram that illustrates that systems biology and altering, perturbations in nodes or edges help to explain how you get from genotype to phenotype, right? Do you remember we finished off there on Wednesday? I had this nice little figure. We had genotype on the left, we had phenotype on the right, and right in the middle was a network where you had a node removed. Well, what protein-protein interactions do, these are these edges, right? These are the edges in our graphs, in our maps. And what protein-protein interactions do in the context of systems biology is keeping it at the same level of the hierarchy of life, take into account things that happen that alter the expression of proteins, but after the transcription, okay? Where you already have intact proteins, but one protein is influencing the behavior of another. And oftentimes not based on altering its transcription, but altering the function of another protein. And it could be because they have the same reactant, they compete for that reactant, or it could be that one protein, if it's an enzyme, the product of that reaction actually inhibits a second protein. And so there could be a number of ways, but it's just, it's an important level and an important application of systems biology. Okay, pathway enrichment analyses. Pathway enrichment analyses. What these are studies in which we are trying to determine two things. One, the significance of an individual node and the determination or the determination, yeah, I guess that works. The influence of a single node in the determination of a genetic illness. Okay, that's one thing we're doing with this pathway enrichment analyses. And another thing we're doing is to try to ascertain the influence of a single genetic illness on a suite of different phenotypes. Okay, so in many ways, what they become is a secondary step after you build a disease map. Okay? After you develop a disease map in which you are taking nodes and you are trying to determine which nodes contribute significantly to genetic illnesses and how and which edges are important, which interactions are important. And then from there, then go and see, okay, given this genetic illness, how much does it influence this particular phenotype or that particular phenotype? And then we can combine these data into a pretty robust study of how several genetic illnesses will all work together. Okay, and you'll see this in a little bit more detail, and hopefully I'll explain it a little bit better when I show you a figure a little bit later on. All right, single target drugs. The remaining key terms that we're going to discuss all deal with how does systems biology improve the development of pharmaceuticals. Okay? That's the rest of these key terms are all going to deal with that concept. How does systems biology make us able to develop better pharmaceuticals? And so the challenge is most of the pharmaceuticals we have are what are called single target drugs. That they are basically designed to target a single biological component, right? To target a single protein, single enzyme, a single receptor, maybe a single RNA transcript, but most often it's going to be some protein. We know from systems biology that proteins do not exist as islands, right? These nodes that are proteins, they do not just have their individual task and then are not connected to other nodes, right? And so when we develop a single target pharmaceutical and we target a specific protein, yes, we may get that protein to behave the way we need it to, but then that impact is going to ripple to surrounding nodes, okay? And so these single use or single target drugs have very predictable and very important side effects that come as a result of targeting an actual component, targeting a node. Because that's what you're doing with this pharmaceutical, right? You are targeting a node, okay? And in many ways, they work almost like you remove that node. And then so everything around it is impacted. And so what we try to do with systems biology is to do what we call network pharmacology. And so if we understand all of the ways in which that specific node, that specific target influences other nodes, we can design pharmaceuticals that instead of removing a node, actually influence an edge, right? And so I want to give you a visual representation of this. Basically had this same thing. Oh, here, let me put one Let me put one right here. I want to create some more relationships here. Okay, so what we have when we're doing single target drugs, we're designing them for a specific protein, which typically these nodes are going to be a specific protein. Okay, so let's just say you have a particular illness that's associated with a malformed version of this protein. Okay, and one of the phenotypic expressions of that is whatever your genetic illness is, right? So you developed a single target drug that basically inhibits this, and so it removes this node. Okay, and so now what you've done is you've eliminated 4 different interactions, and maybe one of those was where you had the disease, right? One of those interactions was the disease, but you've eliminated all 4 of them. And so now you have side effects as a result of removing this node to treat this specific edge, right? Does that make sense? Whereas what systems biology would allow us to do, and what we call network pharmacology, is to better understand all of these interactions so we know that the issue is this edge, right? The issue is this edge. It's not these other edges that this target is involved in. So what we'll do is we'll come in and we'll change this. Because if this, here I need to show this is messed up. Sorry, you need to see that visually. That's messed up. Okay? So instead of saying, okay, well we're going to remove this protein or alter this protein to fix this, we'll just go in and we'll just fix this. Okay? And the way we understand what this is by doing systems biology. Okay? By studying this network pharmacology. Yeah, Alyssa.
What's an example of how you could do that? How can you influence like an interaction by influencing the nodes if they're involved in interactions?
Yeah, so let's use let's use a molecular machine that has a dual purpose. So we've got, what is it? Dehydrogenase, succinyl-CoA dehydrogenase. Anyways, we have one of your enzymes that works in the Krebs cycle as a dehydrogenase enzyme. And so what it does is it removes electrons from the acetyl-CoA molecule. You're basically, you're oxidizing carbon, and you are putting those electrons onto an electron carrier. Okay, so it works in the Krebs cycle to do that. And I think it's succinyl-CoA dehydrogenase. Anyways, but it also is one of the complexes in the electron transport chain that electrons are given to as it goes through that. So let's say your issue that you're having is that enzyme is not properly facilitating the reaction to take whatever its reactant is into whatever its product is and to give those electrons to the electron carrier. But the other role that enzyme plays is still working. It's still acting as a good electron receptor in that electron transport chain. Okay? So one of its issues is malfunction, the other issue is working fine. And so if you go and you modify that protein to get it to work properly in the Krebs cycle, it might very well alter what it's doing in its role during the electron transport chain. Or alternatively, it might be functioning in both of those, but it happens to also act as a hydrogen pump, right? And that's one of the things it does in the electron transport chain is to pump hydrogen ions against its concentration gradient, right? You remember that that's what happens during oxidative phosphorylation? Okay, and so maybe that part of the protein is messed up, and so you design a pharmaceutical to basically alter that portion of the protein, and then it changes its ability to function in other ways as well. And so what we might do is, because let's say we understand that the issue here is the activity of that hydrogen pump, right? That's what's messed up. And then so what we can do is just alter this edge without touching the node. Okay? And you're like, how could you do that? And there are a lot of ways you can do that. You can alter a second protein that isn't as involved in as many things. There are other hydrogen pumps in that electron transport chain that we can target that aren't as involved in as many other things, that don't have as many edges. Or what we can do is change the chemistry a little bit, and there are ways that we can do that. Right? So it might not be as efficient at pumping hydrogen ions, and so what we can do is we can change the chemistry inside the mitochondria to account for that. And there are lots of ways you can do that. But again, and so the whole method of this systems biology is we need to understand these interactions so we can leave nodes alone. Because we nodes, That was nice. We know that these nodes are connected to many other nodes. Okay? So when you touch a node, it influences more than just one edge. But if we can target an edge, okay, now we're targeting a single edge. Okay, and we're not influencing as much of what's going on around it.
But wouldn't the chemistry of the mitochondria also be considered a node?
Potentially.
Stuff like
Depends on what you do. I mean, what do you want to do? Do you want to change the chemistry a little bit so you don't need to work as hard to develop a steep hydrogen ion gradient? And of course, it depends on what you're trying to do. What are you trying to accomplish inside the mitochondria? Yeah. Okay, and then the last thing, palliative drugs. So these are drugs that are designed not to hit a specific target protein, not to hit a specific node, but instead to influence the neighboring processes. Okay? And so these are drugs that aren't designed against a target, but instead are designed to kind of change body chemistry. Or are drugs that are designed to influence something related. And so, again, with these drugs, they're pharmaceuticals that are, again, they're not designed to alter or to eliminate a specific protein that's malformed, but are instead trying to deal with that issue in a different way, without influencing the protein. And if you want an example, it's not a great one, but it's illustrative. Let's say you've got a pain problem, right? And you've got a pain problem because you have a nerve that's overactive. So a palliative drug could be one that just reduces inflammation, right? And so it's not really dealing with that specific nerve and trying to correct what's going on there, but it's reducing inflammation, putting less pressure on that nerve. Okay? That'd be a good, and it's not a great example, but it's an illustrative one of what you might do with a palliative drug. Right, where you're not actually targeting a specific component, you're changing chemistry a little bit. And yes, are there going to be implications of that? Absolutely. You give somebody anti-inflammatories and there are unintended consequences, especially if you're allergic to them. If I told you about my brother and his allergy, okay. That's so funny. It's just, yeah.
Do they more reduce symptoms than actually fix the problem?
Oh, yes. Yep. And you would use them in times where fixing the problem creates unintended consequences, right? So it's like we could fix the problem, but it's going to create other problems. It's going to create other problems. You know what? This is a great example outside of medicine entirely. Let's say your car's leaking oil, right? Your car's leaking oil because you have a small crack in your engine block, and it's like, listen, we can fix your engine block, but your car's really old and that's going to put additional strain on other components in your car, or you can just every couple of weeks put another quart of oil in. Let's just go with the every couple of weeks put another quart of oil in and park over a cardboard box, right? So you can collect that oil and, dispose of it in an environmentally friendly way. That was a pretty good example. You're welcome for that. All right. Any questions on those key terms? Sorry, I usually ask that before I leave our key term slide, but I didn't. All right. So why are diseases comorbid? And so what this is really getting at is what are the mechanisms of comorbidity? What are the mechanisms of comorbidity? And basically, this all boils down to whatever the mechanism is, these are comorbid because they share some node. Okay? They're comorbid because they share some node in the interactome for that phenotype. And that node might be a disease gene. So there might be a gene in there that we know is associated with those diseases, and they might very well be comorbid because they share this same gene. They share the same gene that when mutated alters several different phenotypes. But they could also be comorbid because they share a metabolic pathway. And so what I mean by that is they might both be either part of the same metabolic pathway, like two different proteins in the same metabolic pathway, or the same metabolic pathway might influence both. Okay? And so you see this a lot with metabolic diseases, and so these are usually going to be associated with mutant versions of mitochondrial proteins. Mutant versions of mitochondrial proteins like our dehydrogenase enzyme that also doubles as one of the complexes in the electron transport chain.
Wait, can you encompass into mitochondrial what again?
Mitochondrial proteins. Yeah. Mm-hmm. Although, yeah, and so you can have, most of your metabolic disorders have to do with something's wrong with the mitochondria. You have too many, you have too few, there's something wrong with mitochondrial proteins. There are other metabolic diseases, but most of them, again, have to do with the mitochondria in some way, which makes sense because 30 out of the, on average, 32 ATP generated for glucose molecules require the mitochondrion, right? You can do glycolysis without a mitochondrion, and you get 2 ATP for every one glucose molecule. But if you want to get the 32 on average that you get from every glucose molecule, you need the mitochondria. So most of your metabolic diseases are going to involve that. So if you've got two diseases that are comorbid, chances are they're both metabolic diseases and they're involving the same metabolic pathway. And then finally, the node that they're sharing could be a microRNA. MicroRNAs we've talked a little bit about in this course, but not in a while, so I wanted to remind you of what these are. These are regulatory RNAs. These are RNAs that are transcribed from region of the genome that never get translated. They're non-coding genes. And so instead what they do is they transcribe these microRNAs that target transcripts of other genes and regulate their expression. And so you might have two diseases that are comorbid, not because they share a gene, and this is a coding gene, and not because they're part of the same metabolic pathway or even the same biochemical process or physiological process, but they are comorbid because they happen to be regulated by the same microRNA. And then here are some data from the text, and I thought this was nice. I thought this was a nice little diagram. And so here is a representation of all comorbid diseases. So if you take all comorbid diseases together, like every comorbid disease that we found, doing a phenotypic disease map or a phenotypic disease network, all comorbid diseases together, okay? And we're going to talk about what is the relative risk of getting the second given you already have the first, okay? And if you take them all, that number is 0.15, which means you are 0.15, no, that's 15%. Yeah, you are 15% more likely to get that second disease if you already have the first for all of these comorbid diseases. And again, some of these comorbid diseases are going to be atherosclerosis and type 2 diabetes, Alzheimer's and type 1 diabetes, which we'll talk about in a little bit. Okay, so of all these comorbid diseases, okay, if you take them all, you're 15% more likely to get the second given that you have the first. Now, if the two diseases that are comorbid share a gene, there's some gene that's involved in both, then instead of being 15% more likely to get the second, you're actually almost 30% more likely to get the second. So it's nearly twice as likely that you're going to get the second disease if you already have the first, if the two diseases share a gene. You're like, what? That makes sense, right? But remember, these are complex genetic illnesses influenced by likely dozens, if not hundreds of different genes, and hundreds, if not thousands of different alleles, and a ton of environmental factors. So while that's obvious that you should be more likely, the fact that it's almost twice as likely, that's a little bit surprising. A little bit surprising considering we're not talking about monogenic genetic illnesses here. Okay? We're talking about really complex genetic illnesses. Now, if those two genetic illnesses don't just share a coding gene, but they share a coding gene that is influenced in the same domain of that protein, okay? So the mutation is in the same domain of that protein, and that influences two different genes. Now you're more than twice as likely to get that second disease, given you already have the first, if they share a gene and the mutation that they share happens to be in the same domain of whatever that protein is. Okay. And then this is gene but no domain, so these two together combine to give us this. Okay. Any questions about that? So like makes sense, but it is a pretty significant statement for these complex genetic illnesses. That if they share a gene, that you're now your chances of getting the second, given you already have the first, are almost twice. And then if they share a gene and the mutation's in the same place, more than twice. Yeah.
I still think about all of them. So it's like if you have any genetic illness, the odds of you having any other genetic illness is this percent?
Yes.
Okay.
Yep. You bet. And so this, I don't know, you can say what you want about this and whether or not this is very meaningful, but you need this to compare to this. Okay? All right, so how do we determine comorbidity? So we've got some mechanisms. They might share a gene. They might be part of the same metabolic process if they're metabolic diseases. They might share microRNA. But how do we actually determine these comorbidities? Well, the first thing we do, again, we develop phenotypic disease networks. Phenotypic disease networks. And so these note just observed comorbidities. Remember, these, you're not trying to illustrate any edges, because once you start using edges, now you have to explain those interactions. You're just noting all comorbidities. So if you're like, 70% of people with type 2 diabetes also have atherosclerosis, that's all you're noting, okay? You can talk about, if you want, the next step you can go into like how much of that is environmental, how much of it is genetic, but here you're just noting the relationships. In order to do these and to have reliable data, you need literally millions of data from dozens of different diseases. And so the textbook actually talked about two different, really well done phenotypic disease networks. And one of them was done with like 3.5 million patients and 160 different diseases. Okay? So these are a lot of data that are being used. And again, you're like, how do you handle all those data? And the only way you can handle that magnitude of data is to not try to illustrate why they're related, but just the relationships. Okay? And then you can go to the next step, take two of those diseases and try to ascertain why they're related the way that they are. Because what's interesting, sometimes you do this and you get very surprising relationships, which we'll see in the next slide. The initial construction does not require understanding the relationship. Again, that's a very important point to make about these phenotypic disease networks. To construct these, you don't need to know how they're related. You just need to know that they are related. For instance, somebody was telling me about it. I haven't seen the data. I don't know whether or not this is true. But somebody was telling me about this remote people group that they found inside of the Amazon that are very highly related to Australians. Genetically. Like, I haven't seen the data, so I don't know if it's true. You know, it's a reliable source. I was like, that's really fascinating. They're a remote people group inside of the Amazon rainforest. And they're very genetically similar to Australians.
Have you read the book Kon-Tiki?
No.
It explains why.
It explains why. Yeah. And so is it historically accurate? Yeah. And so, you know, it's like, all we're doing is noting relationships. We're not describing how. Oh, if you look at, this is a good one. This is a really good one. So if you look at some of our mitochondrial genes, we are very highly related to great white sharks.
Nice.
Right? But we don't need to try to ascertain why. Right? I mean, we both eat garbage, right? I mean, we both pretty much just, I'm sure a great white shark would get down on some Taco Bell. But I think it would rather eat a seal that had consumed Taco Bell. But I don't know. Or a person. Yeah, sorry. They actually don't really like to eat people. It's usually accidental when they eat people. And they usually don't eat them.
How do
They
you
just kind of
accidentally
take them. Because you think there's something they're not. Yeah.
Really?
Yeah. Now some sharks are very aggressive. Great white sharks are not aggressive sharks. Okay? They are not aggressive sharks. They just happen to be ubiquitous and you find them pretty much everywhere, and they like to, be in places where humans like to be. Because they like to be in places where there are seals to eat, and humans like to be in places where seals like, because seals like warm waters with lots of food, and we like warm waters. With lots of food. Yeah.
Oh, this is totally not related, but
I love it.
sorry. I heard that actually they realized that sharks don't confuse seals with people because they deal with totally different. So they don't see very well out of water, so they like just chomp to the taste of it.
They don't even see well in water.
Yeah.
Yeah, they really don't. They don't see well at all.
But they don't think that people are seals.
I don't know how much they think. Period. I think they just.
Especially the megalodon.
Yeah, the megalodon. Yeah. I'm always. Is it weary or wary? Weary. I'm always weary of papers that try to explain. Wary, not weary. Wary of papers that try to explain what an animal thinks. Right? Because you'd need I would need some pretty significant data to be convinced that somebody could tell me what a great white shark is thinking when it rams something.
But you can know what it's not thinking.
Maybe. Or you can know that it's not thinking, period. Subsequent studies. So after we've done these phenotypic disease networks, we can then go and say, all right, we're going to take 2 of these genetic illnesses, or 4 because we're ambitious, and we're going to try to figure out what are the molecular relationships among these diseases. What are the systems biological relationships among these? And then what are some of these environmental studies? But again, these have to be second to finding these comorbidities. So, how have these studies been useful? So I want to show you some of these relationships that we found to be very surprising. So here are 5 different autoimmune diseases. And so by autoimmune, it's your immune system is overreacting in some way. It doesn't necessarily need to mean that they initiated the targeting of self-tissue, because it could be autoimmune even if they're targeting something foreign, but their response creates issues in your tissues, okay? So we've got multiple sclerosis, rheumatoid arthritis, Alzheimer's, Parkinson's, and type 1 diabetes, okay? And so there was a group that had done one of these phenotypic disease networks, found out that type 1 diabetes and Alzheimer's happened to be really highly linked. And actually, of all 5 of these are the 2 that are most significantly linked, most significantly comorbid. It's like, that's really interesting, because not a lot of people have published on the role that your immune system is playing in Alzheimer's, okay? Which, this is really intriguing. And this is something pointed out by doing one of these phenotypic disease networks, and then subsequently, researchers have gone in to try to ascertain this relationship. You know what they found also when they were trying to figure out this relationship? And this is kind of interesting, that in people that regularly use anti-inflammatories for any reason throughout their life in their early years, like teenage up to 30 years old, they are significantly less likely to develop Alzheimer's. Significantly. Like 100 times less likely. And you're just like, that's really interesting. You're like, man, I eat ibuprofen like Skittles. I'm feeling pretty good right now. Yeah.
Is there a correlation between type 2 diabetes and Alzheimer's, or just type 1?
Oh, I'm sure there is, but it wouldn't be for the same reason. Because type 1 diabetes has to do with T cells, and so does Alzheimer's, that we know a little bit more about this now. So, but I'm sure there is a relationship and a comorbidity between those.
So I speak of the T cells, not because of the glucose levels in your brain eating your...
Both. Yeah. Yeah. Okay. And so, these comorbid studies, these phenotypic disease networks, they've revealed relationships nobody knew about before. Like the relationship between type 1 diabetes and Alzheimer's. These studies have also informed our research on genetic illnesses, like in the case of Alzheimer's. Seeing a link between Alzheimer's and other autoimmune diseases have encouraged researchers to look into the role that the immune system plays in the symptoms of Alzheimer's. You know, I was very encouraged by this because my wife sent her stuff to 23andMe and it came back, and she has several of the genetic markers for late onset Alzheimer's. And she didn't really worry too much about it. Her mom got freaked out about it, and it just said that because of all of her genetic markers collectively, she's 3 times more likely to develop Alzheimer's after the age of 80. It's like after the age of 80, like that's really late onset, and I don't know if you have to worry about that too much. But my wife eats ibuprofen like Skittles, so I think she's good to go. She's good. Now her stomach, on the other hand, that's another story. But with the Alzheimer's, we might be okay. And I won't be alive when my wife turns 80. So, you know, also not something I have to deal with.
Oh my word.
Sorry, Reagan, you know. All right, finally, these studies have spurred pharmaceutical development. And again, it's going back to the idea that with systems biology, we can develop pharmaceuticals that instead of altering nodes, alter edges. Okay? Instead of altering nodes, alter edges. And you know a really good way to do this? Don't have the target be a protein, but instead have the target be some regulatory element that influences that protein. Okay?
What did you say?
I don't know. I think I said instead of having the target be a protein, have the target be one of the regulatory elements that influence that protein. Influence the expression of the protein, influence the degradation of the protein, influence, any number of things. Okay? Influence how the protein gets modified. Okay? There are a lot of ways in which we can do this. And this is another thing that this systems biology in general has done for the area of pharmacology, but how these phenotypic disease networks have specifically influenced the area of pharmacology. Okay? Because as we understand how diseases are comorbid and which diseases are comorbid, we become better able to predict side effects of pharmaceutical agents, and we also become just better able to design pharmaceutical agents that are going to treat a genetic illness while also preventing another. So that's all I have for you, unless you have any questions. Yeah, Rachel.
Back to the slide that had the different percentages of the likeliness to develop the second disease. I didn't catch how you explained the last.
Oh, this one?
Yeah.
So the gene with no domain, these two combine to get the total gene sharing. So this is like, if you ignore these two columns, this is all diseases versus diseases that share a gene. And then these two are like, okay, given that they share a gene, if they share a gene where the mutation's in the same place, you're more than twice as likely. If they share a gene where the mutation is in a different place, they're less than twice as likely and less than what is overall. Okay, and the fact that this bar is closer to this bar than it is to this bar gives you an indication that a lot of times these comorbid disease sharing, the mutations are in separate places.